welcome. This is the Association for Creatine Deficiency's Parent Education webinar series on how to read your genetic test report. I'm Erin Collar, Director of Communications with ACD and mother to a 6-year-old boy with creatine transporter deficiency. Understanding my own son's genetic test report and variant information has been a journey over the past 4 years since his diagnosis, and I still have a lot to learn. As research continues to progress in the search for better treatments and a cure for CCDS, I'm learning that it's more important now than ever to know what kind of mutation my son has and learn as much as I can about it. We are aware of over 100 different genetic mutation variants for CTD, just to give an example. And right now there are 2 collaborative ACD-funded research studies that are currently seeking customized treatments for CTD. That means variant specific or mutation specific. And it's likely that treatments for GAMT will follow the same path. Also, there may be more understanding about different severities of CTD, AGAT, and GAMT based on data gathered from these mutations and the patient registry. An upcoming Creatine Decoded article will dive into the details of these studies, which are working on understanding SLC6A8 mutations, as well as how each individual mutation may respond to a variety of drug compounds in lab tests on cells. So as caregivers, we can become more actively involved in the process of finding treatments for our loved ones, and that begins with understanding their diagnosis. So I'm pleased to introduce today Julianne Savat, who will help walk us through everything we need to know about genetic reports and types of mutations. Julianne Savat received her Master of Science in Genetic Counseling from the University of North Carolina at Greensboro in 2015. She is a board certified and licensed genetic counselor who has served patients in the Geisinger Autism and Developmental Medicine Institute and Cancer Genetics Clinic in Pennsylvania. In terms of her research interests, she is motivated to empower patients to be active partners in genomic discovery and is working towards that goal through being part of the ClinGen patient data sharing team. Please share any questions you have today in the Q&A section, not the chat, but the Q&A right next to it at the bottom of your screen. And Julianne will be available after the presentation to answer your questions. This webinar is being recorded and it will be shared on the ACD website, but the Q&A portion will not be recorded. So with that, I am happy to pass this along to Julianne for her presentation. Thank you.
Thanks, Erin, and thank you to the ACD for having me today. I'm really delighted to get to speak to you all. As Erin mentioned, I will be providing an overview of some of the common terms that you might find on your laboratory report. For some of you joining us today, I imagine that some of these concepts and terms might be familiar. For others, they might be new terms to you. So my hope is that we can review concepts like chromosomes, DNA, and genes, discuss the types of genetic changes or what we call variants, types of genetic testing, and some other common terms that you might come across when trying to decipher your child's genetic test report. We'll also walk through an example report, and I welcome you to pull out your report if you happen to have it handy and want to keep it as we're walking through the different terms and pieces that you might see on there. In addition to walking through how to review your genetic test report, we'll also spend a few moments discussing the ACD registry, as well as the option to participate in data sharing through that registry. As Erin mentioned, we'll end with that Q&A, and this is my plug to remember to use the Q&A box as well. And feel free to type those in as I'm talking, so as you think of questions. So first, over to how to read your genetic test report and some of the common terms you might see. So all of you listening, or many of you listening, have likely gone through the genetic testing process and might have spoken to a genetic counselor or a geneticist. So as I said, some of this might be familiar. But on the screen here is an example report we'll be walking through. You can see there are many terms like X-linked, and there are letters and numbers, words like de novo, and I hope to walk through each and every one of these terms today in hopes of helping you understand just a bit more of what that other language is on that piece of paper. So first, a bit about cells, chromosomes, DNA, and genes. This may be a review, but I like to sort of set the stage and work from there. Each one of us has trillions of cells that make up our bodies. We have different types of cells, including our skin cells, brain cells, heart cells, They're all like our building blocks. In each of our cells, all of our genetic material or our DNA can be found. Our DNA is sort of like our body's instruction manual, and each individual instruction is called a gene. To keep our DNA organized in our cells, it's wrapped up really, really tightly into these structures called chromosomes. Typically, people have 46 chromosomes that come in 23 pairs. Our chromosomes come in pairs because we get one set from our mom, the other set from our father. The first 22 pairs of chromosomes are the same in males and females and are just organized based on size. So we start with the largest, number that 1, all the way to 22. The 23rd pair of chromosomes are called our sex chromosomes, and males are typically XY, females are typically XX. As I mentioned earlier, each of these chromosomes is made up of DNA that's wound up very tightly, almost like a ball of yarn. The yarn's the DNA, the ball itself is a chromosome. Our DNA provides instructions for how our body makes up proteins. And like I said before, each instruction is called a gene. We have approximately 20,000 genes that we inherit from our mom and dad, and they help our body grow, develop, and work. Each gene has a specific role within the body. Some do things like determining our eye color, some help our heart develop, some help synthesize and transport creatine. So for example, the genes associated with cerebral creatine deficiency syndromes code for proteins that are important for that creation and transport of creatine. SLC6A8 codes for the protein transporter that takes creatine to the brain and muscles. GATM codes for the protein that's important for this first step in creating creatine. GAMP codes for the protein that takes another compound and breaks it down into creatine. So they each have a role in the body. So how does our body read instructions in our genes to make proteins? Our body actually spells out the instructions in our genes using the letters A, T, G, and C. And that's what our body reads to make those proteins. When reading those letters within our genes, our body actually reads the letters in sets of 3. A set of 3 letters in our DNA codes for 1 amino acid, which is just the building block of protein. So each of these is amino acid, the whole thing's a protein. And a set of 3 letters, A, T, G, would code for 1 amino acid, CAC for the next, and so on. There are 20 different amino acids that ultimately make up our protein. Again, all our proteins, all are coded by 3-letter sets of our DNA. So if we look back at our sample report, the first box notes the gene. So this individual's testing found a genetic change in the SLC6A8 gene. So we'll move through the rest of the boxes, but we've talked about that one so far. So now that we've talked about DNA, genes, and proteins, let's discuss genetic variants or mutations or genetic changes. These words all mean the same thing, different ways to describe it, but they're just changes in those letters of our DNA. While most of our DNA sequence is the same from person to person, we all have genetic variants. They're part of what make us unique in ourselves. Some variants can affect how proteins work in our body and can cause developmental or health differences. There are 2 main categories of genetic variants that I want to mention or sort of focus on today: copy number and sequence variants. So first, copy number variants. Copy number variants, or CNVs, are genetic changes where a person has often thousands of extra or missing letters within their DNA. There's large missing or extra pieces of genetic material. If a person is missing genetic material, we call this a deletion. If they have extra genetic material, we call this a duplication. So if we think about our genetic material, our DNA, like a shelf of books, a copy number variant is like having a missing or extra chapter of that book, or maybe half of a book. So it's a large extra or missing piece. Copy number variants, if you see these sorts of things on your report, they're often named for their location along those chromosomes that we talked about. So you might see a report that says that there is a deletion of 19p13.3. This tells us a couple of things. This tells us that the person has a copy number variant first on the 19th chromosome. When we talk about chromosomes, we divide them into 2 parts. what we call the short arm or P arm, and the long arm, the Q arm. So this P in the name just tells me, okay, what part of the chromosome are we talking about? It means the short part. From there, the 13.3 of the 19p13.3 tells us exactly what stripe of the chromosome we're talking about. If we look at chromosomes under the microscope, they have this striped pattern. So it tells me, okay, what piece on that short arm is missing or extra. So if you have a report that has 19p13.3 deletion, that's what that means, that there's a missing piece on that part of the chromosome. The other type of genetic variant that I think many of you have questions about and probably is more applicable to your group are sequence variants. So I'm going to spend much more time on these today, but I didn't want to leave out copy number variants. I wanted to make sure we mentioned them as well. So sequence variants means that there's a change to the letters within a gene. Sometimes this is a single letter that's different. Other times it might be a few letters, but it's just a small number of letters that are changed. If we think of our genetic material as a shelf of books again, a sequence variant is like there being a spelling change in one sentence of the book. Sequence variants are typically named based on the change to the DNA and the change to the protein. So if you see lots of C's, P's, letters, and numbers, those are telling you what's happening to the DNA and what's happening to the protein, and we'll walk through each of those. If you see a C dot on your report somewhere, so a C period, like here, that means that this is describing what's different in the DNA. So in this example, we're talking about the SLC68A gene, the DNA. The numbers tell us what letter are we talking about in that DNA, in that gene. So here we mean we're talking about letter 928. The first letter that you might see after that number tells us what DNA letter do people typically have at position 928. In this example, people typically have a G. The greater than symbol then means sort of our way of saying is changed to. And the letter after tells us, okay, in this individual, what letter are we seeing there? So all together, this example tells us that in the SLC68A gene, at position 928, if I start at the first DNA letter and count all the way up, most people have a G. This individual happens to have an A. That's what's happening to the DNA. As I said, the genetic change can also be described by, or the change can also be described by what happens in the protein. So if you see a P period on your report somewhere, this is describing what's happening to that amino acid change, that protein. This P.GLY310ARG tells us that at amino acid 310, so if I have that protein all stretched out, start counting at 1 all the way to 310, most people have an amino acid called glycine. This is an abbreviation for it. So this position is what's typically there. And then the second abbreviation, arginine, is what this individual has. So because of that change in the DNA that we talked about, the amino acid 310, that's typically a glycine, has been changed to an arginine. So there are several types of sequence variants that I want to mention and talk through, since you might see these terms on your lab report, or your C dot or P dot might not have matched up exactly with that example. So I want to make sure we walk through these as well. So some of you might also have questions about, does my child's variant cause a misfolded protein or a shortened protein? And hopefully walking through some of these examples can help you sort that out from your report as well. So the first type of sequence variant that I want to talk about today is what's called a missense variant. Missense means that one amino acid, one of those protein building blocks, is substituted for another because of the change to the DNA. So in the example that we just walked through, that amino acid at position 310 was the only thing changed, one amino acid. So this is an example of a missense variant. A single amino acid change might change the function of the protein due to changes to folding or other things. Another type of sequence variant you might see is what's called a nonsense variant. So like missense variants, nonsense variants are caused by a change in a single letter of the DNA. So here we see at position 522, typically a G. In this individual, it's an A. But in addition to coding for amino acids, the 3-letter sets that in the DNA that our body reads also tell our body when to stop making the protein. So certain 3-letter sets say, stop, all done. So in a nonsense variant, that happens when the DNA change results in an early stop signal. So before we would expect that stop signal to happen. This makes the protein shorter than typical, which means it might not be able to function as we would expect. If you're looking at your report and you see things like TER, termination, an X or a star in the protein description, these are ways of noting that this is a nonsense variant. You also might see sequence variants that result in a small number of extra or missing letters within the gene. Insertions and duplications of a small number of letters add letters to the gene, and deletions remove a small number of letters to the gene. In the example here, this is a duplication. The dupe in the C dot tells us that this is a duplication of letters. And you can see that there's typically the sequence AAC in the gene. And in this individual, there's an extra one. We see that set of letters repeated again in the sequence. Dupe, as I said, often is used to make note when there's a duplication. INS can be used if there's an insertion of letters that aren't typically in the sequence. And then for deletions, DEL is often used. So in this example, there's a deletion of 2 letters. Letter 1000 and 1001 are typically A and T, but they're removed from the sequence in this individual. Adding or removing letters in the DNA sequence can result in 2 different changes to the protein, types of changes to the protein. The first is what we call a frame shift variant. So as we talked about earlier, the body reads the DNA sequence in sets of 3. We call that sort of our reading frame. If we duplicate just 1 letter or a few letters, not in a multiple of 3, that shifts our body's reading of the rest of the gene sequence. So we're not able to read it in the same sets of 3 anymore. It sort of shifts how we're looking at things. You can identify frame shift variants because they typically have FS in their protein description. And ultimately what happens is it shifts the reading frame, changes some amino acids, and then eventually we get a stop codon somewhere down the line. So this tells us that this duplication results in a frame shift. The protein kind of goes along, looks different than typical, and 26 amino acids down the line, there's a stop. Adding or removing letters in the DNA sequence can also result in what we call in-frame changes in the protein. So if we have that 3-letter reading frame and we remove 3 letters within that frame, that will just remove a single amino acid in our protein. The rest looks the same. We haven't shifted how we're looking at things the rest of the way down the protein, but we have changed the protein sequence by removing an amino acid. Our body's still able to read those remaining letters, but like I said, we have this change. So you might see if there's a deletion in the DNA, if it's in frame, you'll just see a deletion in that P dot, telling you that an amino acid or maybe a few amino acids were removed because of that DNA sequence difference. So let's look back at our example report. So on the report, we see a box with variant and a box with coding DNA. If we look at what's in those boxes, we can better figure out, is this describing the protein? What is this about? So in the coding DNA box, we see that little C dot. So that tells me we're describing the DNA change here. We see, I made it a little bigger down here, C.1222, and to a little dash, 1224, del TCC. So these numbers here tell me that we're talking about 3 letters in the DNA sequence. 1222, 1223, and 1224. The del tells us that we're talking about a deletion. And then these are the 3 letters that are impacted. So taken together, at letters 1222 to 1224, most people have a TCC, and in this individual, they're deleted. So they're missing from that sequence. The variant box has a P dot, so we know that this is describing the protein. Again, the number tells us what amino acid are we talking about in that protein. If we start counting at 1 and go up, we're talking about number 408 in this case. The first letter, and it's typically an abbreviation for some amino acid, you might see 1-letter or 3-letter abbreviations, is the reference, what's typical at that position. So in this case, this is typically a phenylalanine at position 408, and the del means that it's deleted or missing from the protein sequence in this individual. I know these are a lot, and we can talk through more examples and questions, but hopefully these little pieces have been helpful so far. And to tie it back to what that example of sort of frame shift in frame, this is what we would consider an in frame deletion, because it changes just 1 amino acid, removes 1 amino acid from that protein sequence. So now that we've talked about genetic variants, sometimes I find it's helpful to talk at least briefly about types of genetic testing and what sort of variants they pick up. So sort of walk through what would have identified these types of changes. All of you on the webinar today are likely familiar with 1 or more of these tests from your own genetic testing journey. And I think, as you know, these are typically done using blood or saliva samples. The first testing type I want to mention is a karyotype. A karyotype is a test that we used quite frequently some time ago, and it looks at the chromosomes for large copy number variants, things we could actually see under a microscope. With this testing, we can identify really large or missing extra pieces of genetic material. If we zoom in a bit further, the next test we can do is microarray, and this can identify smaller copy number variants. If we want to detect sequence variants, the next test we need to think about is sequencing. With this testing, we're actually looking at the individual letters of a person's DNA sequence and seeing if we expect an A there, what do we see there? There are different types of sequencing that's done. Some people might have single gene sequencing. Some individuals might have a gene panel that looks at for variants in a list of specific genes that are tied to certain health concerns. And then some individuals might have exome sequencing, which uses sequencing to look for variants in all parts of our genes that provide our body with instructions, or what we call exons. So if we look back to our report, we see this individual has had exome sequencing. The testing is often noted at the top of your report. So if it's been a little while and you're just curious which one of those tests your child had, you can look for that on your report as well. So when a genetic testing lab identifies a genetic variant through testing, the next step is to interpret that genetic change or try to determine if it impacts how the gene works in the body, if it's associated with a health condition, developmental difference. As I mentioned earlier in my talk, we all have lots of genetic variants that make us unique, but some do impact our health. So when a lab sees a genetic variant, they try to use all the information available to them to try to determine if it's associated with a particular condition. For example, this means looking at how the variant impacts the protein. What is the change to that protein? Where is it located in the protein? They look at the literature to see if the variant's been seen in people with symptoms. They look to see if the variant's been seen in healthy individuals, and then put all of this information together to assign an interpretation. These interpretations or classifications fall into 3 main buckets: benign, uncertain, and pathogenic. Benign means that the copy number or sequence variant that they found isn't known to cause any significant health or developmental differences. Pathogenic means it's a copy number or sequence variant that's known to cause symptoms or features. You also might see terms like likely pathogenic or likely benign on your reports. And those are just sort of used in instances where there's not enough information to put a variant definitively as benign or pathogenic, but we are almost certain that those are in those categories. If there's not enough information at all to say it's pathogenic or benign, labs will use this classification category called uncertain. This means that there's really not enough evidence to say that the variant is associated with health or developmental differences at this time, and really just more information is needed. And sometimes that might be testing other family members like parents. Other times it might be just needing to wait for more information to come available. So if we look back to that report, we see that this variant on the report that we've walked through was classified by the laboratory as pathogenic. So again, that means that this sequence variant is known to cause health or developmental differences. Other terms you might see on your reports are autosomal dominant, recessive, and X-linked, and I want to walk through these. Before I do that, I wanted to return to our chromosomes just as a reminder. Again, we all have 2 copies of the first 22 chromosomes because we get 1 in each copy from mom and 1 in each copy from dad. For all the genes on these chromosomes, we typically have 2 copies, 1 on each chromosome. Our last set of chromosomes are our sex chromosomes, and again, females typically having 2 X's, so 2 copies of all the genes on the X chromosome, boys having an X and a Y, so 1 copy of the genes that are on the X and 1 copy of what's on the Y. Autosomal dominant and recessive are used to describe conditions caused by changes to genes on those first 22 pairs of chromosomes. So the ones where we all typically have 2 copies, 1 from mom and 1 from dad. Conditions that are caused by having a pathogenic or likely pathogenic variant on just 1 copy of the gene are what we call autosomal dominant. You only need to have 1 genetic change to cause that condition. If pathogenic variants are needed on both copies of the gene to cause a condition, this is called an autosomal recessive condition. So GAMP deficiency is one example of a condition that's inherited in a recessive pattern. Conditions that are caused by pathogenic or likely pathogenic variants in genes on the X chromosome are called X-linked. And symptoms can differ between males and females, since females typically have 2 X chromosomes while males typically have 1. Sometimes autosomal recessive, autosomal dominant, and X-linked are called inheritance patterns, but they don't tell us if a genetic variant was actually inherited from a parent. They're really just telling us how many genetic changes need to cause this condition and was the particular variant found on the X chromosome. But it doesn't tell us a lot about that individual and what's happened in their family. So if we go back to our report, some will have the mode of inheritance. And in this case, for SLC68A, it is an X-linked condition because it's on the X chromosome. The words heterozygous, homozygous, and hemizygous also might be on your report, and this tells us about the specific patient that was tested. If the genetic variant was found on 1 copy of the gene, this is called heterozygous. Homozygous tells us that the person has the change on both copies of their gene, and hemizygous is used to describe males who have the genetic change on their only X chromosome. So if you see these on the report, this helps you know what is actually going on in your child, what did they find and for them, and was the variant on 1, both, or on their X chromosome. So if we go back to that report again, this is often called zygosity, that heterozygous, homozygous, hemizygous. And we see here that it's listed as hemizygous. So this particular gene, SLC68A, is on the X chromosome, and this is a male who has the change on his only X chromosome. Some genetic changes are passed from parent to child, and those are said to have been inherited. So if you see words like inherited on your report, if parents were tested, this tells us that it's been passed from a parent to child. As many of you might know, some genetic changes are new and unique to a child, meaning that they weren't inherited from a parent, but happened early on in development, maybe in just sperm, egg, early on in that child's development. We call these de novo, which it means anew. So it's new and unique to the child. For de novo variants, oftentimes these are things that happened early on in development, but occasionally these are things that there is a chance for future children to have because maybe multiple eggs or multiple sperm of mom or dad had the same genetic change, which we'll talk a little bit more about in a second. So going back to our report, there's a little box that said inherited from, and we see de novo written there. So this means that this genetic change is new and unique to the child, tested mom and dad samples, and they did not see the genetic change in them. Finally, a few families who are listening in might have the words mosaicism or mosaic listed on their report. So mosaicism means that only some of a person's cells have the genetic variant. This means that some cells have the variant and others do not. And for some families, this might be identified through their testing. Not all testing picks up mosaicism. You need to have that genetic change in the particular tissue or cell that's being tested. Our testing doesn't pick this up all of the time. But when it is, again, it means some of the cells that we tested had the genetic variant and others did not. With our current testing technology, we often can't tell which cells have the genetic change in the body or do a good job of knowing how many cells in the body have the genetic change just based on the information from our testing. Sometimes a parent might have a specific type of mosaicism called germline mosaicism. And this is that instance where some of a mother's eggs or some of a father's sperm cells, what we call their germline, have the genetic variant. Our current technology doesn't allow us to detect germline mosaicism. We don't have testing for it, but it is usually identified when parents have more than one child with the same genetic variant, but don't have that variant detected through their blood or saliva samples. If a variant is thought to be de novo in a child, the risk for this varies sometimes from condition to condition. Many conditions are a 1% chance. Some are higher depending on that particular gene and condition though. And the chance that family members will have the same genetic change really does depend on the inheritance of the pattern of the condition and whether the variant was inherited or de novo. So I know this is often a question about recurrence risks and what are the chances, and sometimes that's specific to what exactly is on your report and what was found. So with that, now that we've walked through, probably quicker than we should have, all of the terms that can be on your report, I want to turn it over to Sophia, at least briefly, to talk a little bit about the registry, and then we'll talk quickly about data sharing and its importance.
Thank you so much, Jules. I look forward to the Q&A. If you have more questions, please leave them in the Q&A. We'll be doing the Q&A in just a couple of minutes. So how ClinGen and ACD are connected? So we are using our new creatine info registry. We launched the registry back in March, and this will be the platform that will let us connect the ClinGen data share program with the genetic reports from the community. So if you log in, if you register to the registry, you can log in as a participant, meaning the person with the CCDS diagnosis, or as a parent or caregiver or legally authorized person on behalf of the participants. Next, please. You'll see that in the initial surveys, thank you, Jules. In the initial surveys, we have only 5 surveys. One of them is diagnosis. That is a place where you will be able to upload your genetic report. And then we have another survey called ClinGen Data Sharing Informed Consent. Next one. Instead of the diagnosis survey, you will have the chance to upload your genetic report. If you don't have your genetic report now and you may have it later, you can click on yes, and I will upload it later. We are preparing a medical report survey that will come soon, where you will have another chance to upload your genetic report. And if you need help getting your genetic report from your doctor, this is something that we can help you with. And later, Jules will share with you how. Next one. The ClinGen data sharing informed consent has more information about the ClinGen data share program and will give you the option to opt in to give access to ClinGen to your genetic report. And we'll ask you if you would like to get contacted by someone from ClinGen to receive updates, potential updates of your genetic test results. If you have any question about the registry, I'm always available for you. Really happy to hear from you. So please reach out at registry@creatininfo.org. And now Jules will share what is the data share program and why it's so important for our community.
Thanks, Sophia. And I will try to move through these extra quickly because I see some great questions in the Q&A, and I want to make sure we have plenty of time for that. So I think you've heard that I'm a part of ClinGen, this ClinGen data sharing program, and I thought it would just be helpful to tell you a little bit about ClinGen to start. So ClinGen is an NIH-funded resource that's working to better understand genes, variants, and their relationship to health, essentially. ClinGen recognizes that data sharing is an important piece of that we need to have information available to better understand these things and better understand genetic conditions and rare disease. Because of that, ClinGen encourages data sharing from several stakeholders, including genetic testing laboratories, genetics researchers, genetics clinicians. But for some time now, going on 7 years, we've also encouraged data sharing from patients, and in particular, data sharing with a database that's called ClinVar. So our team is working with individuals, advocacy organizations, and registries to share their information with this publicly available database that holds de-identified information about genetic variants so that it's available to the healthcare providers, labs, and researchers who are using it, who are caring for patients, who are doing testing, and who are researching genetic conditions. This database is public and freely accessible, and it is largely used by clinicians, labs, and researchers. The database really relies on people sharing their data. So it relies on labs and clinicians and others who are choosing to submit information. Ultimately, data sharing is important because it can help increase our understanding of genetic conditions and can help doctors and genetic testing labs better understand genetic changes that cause disease, health differences. This increased understanding can ultimately help identify interventions and treatments, also can help individuals get clear genetic test results. And really our hope is that the more information we collect, the better we will understand how genes affect health and ultimately how to best care for patients. So that's ClinGen's goal and why we're working with patient groups, patient registries such as ACD to share data. As Sophia really summarized well, participants in ACD have the option to participate in data sharing with ClinGen. So this is an optional piece. You don't have to register elsewhere. It's just an additional survey within your registry. You enroll in the registry as typical and would complete that consent survey if you are interested. This consent walks through how de-identified genetic and health data are shared. So again, it's de-identified, your name and other identifiers aren't ever shared. And it does allow a small number of our team, so myself and one other genetic counselor, to access your records to pull out the important pieces from your genetic test report. So what we walked through today, we walk through and pull out the variant information, the classification, so that we then can share that information with ClinVar. As Sophia mentioned, if you no longer have a copy of your records, our team can help request one from the genetic testing laboratory that did your testing or your healthcare provider. And then from there, once we have the information, we de-identify it and share both the genetic information and health history information with ClinVar on behalf of ACD, which makes it accessible to those other stakeholders that we talked about. I'll briefly just mention a couple of benefits of data sharing that we've seen from the groups that we've walked through so far. So high level, I kind of walked through, we hope this increases our understanding, but we've been engaging registries in data sharing for over 7 years now. And from our experience, the first benefit that we've noted is that patients who are sharing data often share more information than what labs or researchers or clinicians share. So the information that we submit has those health details, can have inheritance of the variant. And sometimes that can be really useful in better understanding genetic variants and genetic conditions in the future. Labs often have this information, but it's just not part of what they typically share with the database. So patients are providing more details, which can be really useful. In addition to providing additional details, we've also seen that 41% of the genetic variants that we've shared so far have been new to ClinVar. So that means that the reporting lab or another submitter hasn't put it out there in the public space before. So if that family didn't take the time to share their data, their variant wouldn't have been in the database otherwise. And this is really useful the next time a variant is seen or similar variants are found in other patients, or if a researcher wants to connect back with ACD to talk about additional research, it's helpful. I'm going to skip a few slides just in the interest of time. In addition to contributing de-identified data to ClinVar, one other piece that you'll see in that ClinGen patient data sharing program survey is the option to receive updates about your genetic test results. So genetic variants are classified when they're reported, and over time, our understanding might shift. For example, your report might say variant of uncertain significance, but that was from 2012. And in the meantime, more information's come available, and now that laboratory thinks that it's a pathogenic change. Because we can become aware of some of these things through our data sharing, we do give participants the option to receive these updates through their participation in data sharing. Sometimes updated information might be shared back with patients by your doctor or the lab, but we know that doesn't happen all of the time. And so this is just another way that you can choose to receive updates and learn more moving forward. Now I'll end just quickly with what this looks like. So one question I get a lot is, well, what does my information look like in ClinVar? So these are screenshots of a ClinVar variant that was submitted on behalf of ACD, someone who chose to participate in data sharing. And each page in ClinVar has the variant. So we talked through much of these things like that C dot and P dot today. And if you scroll down, it has each of the individual submitters. So here you can see that this was submitted on behalf of ACD by our team. And you can see some of those details like inheritance, how it was interpreted by the reporting laboratory. In this case, likely pathogenic. Most likely it's associated with a health difference when it was reported. You can see additional information, health details on the next page. And it's just things that that person has selected yes to on their diagnosis survey or their health survey. You also see age range and then the sex that the parents provided. But again, no identifiers like name or date of birth or anything like that. Thank you so much for having me, Erin, Sophia, and others. It's been really a delight to be here this afternoon, so thank you.
Thank you. It was so informative. We loved having you share all this with us. Thank you so much.